Now, the scientific method is what's termed a deductive process. There's two types of reasoning: inductive reasoning and deductive reasoning. Inductive reasoning is where you can use observed phenomena and predict future events. All right, observed phenomena, right? Did the sun come up this morning? Yes. You didn't see it, but it's definitely up. Did it come up yesterday? The day before? The day before? The day before? There's a pattern of sunrise. So what's going to happen tomorrow? The sun's going to rise. That's inductive reasoning. We're looking at a pattern, and then we're going to make a general statement about it. Deductive reasoning goes in the other way. You're going to test things. You're going to come up with a proposed solution and then test it, kind of hypothesis. And then you're going to use observation and then confirm your original description of the world. So inductive reasoning moves from observations to seeing a pattern to forming a theory. The sun will rise because we've seen the sun come up again and again and again. Whereas deductive reasoning starts with a hypothesis. This will happen. And then you test it, and then that confirms the hypothesis or refutes it. So the scientific method, what we're going to deal with is deductive. We don't use much induction in biology. You're going to use a lot of induction in sometimes in psychology, a lot in sociology. Now, I do want to point out there's a difference between a scientific theory, a scientific law, and the modern vernacular theory. A scientific theory is an explanation that's been based upon supported hypotheses and verified many, many, many times. A theory is our absolute best understanding of how the world works right now. It's not just a random guess. Common parlance, when people are normally just talking, they're going to say things like, I have a theory about that. And when they say that, what they mean is, I have a guess. I have an educated guess, but it's just a guess. Scientists don't use theory in that way. A theory is our absolute best understanding based on lots and lots of evidence. So that's a theory. It is really, really been shown again and again. Theory of evolution, theory of natural selection, theory of gravity, yeah, right, theory of relativity, theory of oh shoot, remember those times where words just disappear? Theory of inheritance, chromosome theory. We have lots of theories, and they are our best explanation as to how the world works. That is not how most people use the word theory, but it's how scientists do. The other thing we'll talk about is scientific law. Now, a scientific law is the cornerstone of scientific discovery. A law is simple, true, and absolute. It's universal. It's everywhere. If a law did not apply, then all science based on that law would collapse. Physicists love to use laws. They always talk about laws. At least they used to. 1600s, 1700s. What are a few scientific laws you've heard of? Here, I'll say Newton's laws of gravity. Okay, Newton's laws of motion. There's all sorts of laws out there. The problem is we're starting to realize that humans don't know everything. Right, where laws need to be universal. It's Newton's laws of motion. What's in motion stays in motion until acted on by another object. Works just fine under certain circumstances. But we don't know how it works in the middle of a black hole. Right, no, there's all sorts of things we just don't know. So we're starting to understand there's a lot we don't know. So you don't get many laws nowadays. What you get now are theories. And again, scientific theory, not common parlance theory. Now, in order to discover new theories, we're going to use the scientific method. The scientific method is awesome. Let me tell you the history of the scientific method really quick. This is fun. There was this guy, history. Any of you guys going into history? No, who's going into history? Back in the late 1500s, there was a gentleman explorer by the name of Sir Francis Bacon. Cool name. He wrote poetry. He was a fighter. He was and he was a scientist. And he came up with this method, the idea of you make observations about the world. You come up with a question and formulate a hypothesis, your best guess as to how it works. A statement, a falsifiable statement. You develop predictions based on that statement, and then you test your predictions. And if your prediction is, if the test proves that your hypothesis is correct, or it supports your hypothesis, then you move on to develop the general theory off of that. But if it's not correct, if it shows that you're wrong, you sort of go back, you refine, you alter your hypothesis. It's a self-corrective cycle. And so he put this whole thing together, and do you know what he called it? Take a wild guess. You'd think scientific method, right? Scientia, though, is not English, and he was an Englishman. He called it the Baconian method. Because scientists, if you learn nothing else, love to name things after themselves. The Baconian method. And then the people in France, the French rationalists, Descartes, you guys have taken philosophy. Okay, good. Well done. Keep it up. The French rationalists, Spinoza, Descartes, Leibniz, they turned around and said, nah, that's an English word. We're going to call it science. And the science. Scientia, knowledge. So we're going to work on, I'm not going to go into like, you have to memorize the steps of the scientific method. You've been using the scientific method your whole life. Whether you know it or not, you have. Any of you guys have brothers or sisters? Okay, so a bunch of you guys do. You're like, no siblings? You do? Brother or sister? Brother and a sister. What kind of car did your parents drive? Right now? No, when you were younger. Oh, wait, do you still drive in the car with them? Okay. And your siblings? Really? Good for you. Okay, your siblings in high school? Yes. Okay, this is even more fun. Now, what kind of car do they have right now then? I don't know. You're in high school and you don't know what, okay, how about this? Is it a little car or a big car? Little car. How many seats are there in the back? 3 seats. Now, is it on a bench? Do you ever have to ride with your siblings? Sometimes. When you were younger, did you have to ride like we were all squished together? Yeah? Do you guys remember this? Yeah, you've been there. All right. What's your name again? Anna. Okay. And you're Annie. Okay, sorry. I'm trying to work on it. Okay. You got your siblings. Were you in the middle or were you on the side? You were on the side. You were the lucky one. You weren't in the middle? Sometimes. Sorry. If you have siblings, you know exactly what it is that sets them off. You know in your heart of hearts how to get them in trouble with your parents and you look good. Right? All right. Do you guys ever play in the car seat? There was your section. You had the window. I assume you had the right window. Left window? I don't even remember. How do you not know these things? All right. So you're in the window. Not in the window. Next to the window. You got a kid here. Now, did you let them touch you? No. There was a buffer zone. And it was guaranteed. And what happened when they crossed that buffer zone? That's right. Something happened. Now, I guarantee you, though, you knew exactly how close you could get to your brother or sister without them swatting you. Didn't you? You could. And you went right to the line. And there were some times you went over the line just to see if you could. And you tested and retested and tested again. Does that make sense? You guys have done this. You know exactly how to bother your siblings because you did it over and they were your biggest test subject. You tested them all the time. Right? Do you guys know what a wet willy is? Okay. Stick your arm around like this and you stick it in somebody's ear. Most people flinch, don't they? Yeah. When do they flinch? What? Once it goes in their ear. You test it again and again and again. What was your name again? Yeah. Kevin. Welcome to the front row. All right, check this out. I am actually going to put my finger in your ear if you let me, by the way. I don't say. Okay, so apparently I can get what? I'm not putting this is the other finger like that far. Okay, ready? It's cool. Okay, still about the same. It's cool, it's cool. So it's the idea you can repeat these things over and over and over again. And you find out the distance. You test and you retest and you test again. You've been using science your whole life. Kids use science all the time. Put your hand on the one time, right? But you still, you do it. You test and you test and you retest. So here we're going to apply the scientific method. Oh. So close. All right, so we noticed something. What do you notice? Yeah, one's missing a leg, one's got too many legs. There's something going on. You've observed this. Step one, observation. What do you do if you see a frog leaping across the road with extra limbs? What? You take a picture of it. That's right. Click. Post it all over the place. You've got your phone, you may as well take a picture. That's cool. You took a picture of it. Life's good. Probably what's going through your head, even if you'd stopped and you're just like, oh, picture. You're thinking to yourself, why the hell does this frog have extra legs? There are lots of possible reasons. Give me well, we'll get into that in a second. But the question is, how or why? Why does the frog have so many legs? What are you interested in learning? So step one, make an observation. Step two, question. Step three, do some research. Again, real life, frog, multiple legs, hopping across the road. You see it, you took your picture. What's the next thing you do? You ask the question. After you ask the question, you don't take a sample. What do you do? He has enough legs to spare? No, come on, guys. What do you really do? All right, you see a frog hopping across the street. You take a picture because super cool, we can do that. You ask yourself, why does this frog have so many legs? Then what do? Do you go on with your life? You look it up. Where do you look it up? That's right. You have your phone out. You just took a picture. So now you Google it. You go to the John Tyler website. Find something from the library. Look it up. That's the right answer. Not what I would have done. I would have absolutely gone to Google. You find out a possible explanation. You come up with a hypothesis. That's the answer right there? All right. With your hypothesis, what is a possible explanation for this? Mutation. Sure, why not? Genetic mutation. Oh, was this the one that didn't work? Yeah. Genetic mutation. Give me one more hypothesis. That's it. Everyone's like, that's all it could be. Mutation. It's a weird freak frog. Evolution. That's genetic mutation still. Mutation. Anything that's not a mutation. Ooh, conjoined frog twin. That's not really a mutation because that's not heritable. Conjoined frog twin. I like that one. Anything else? Science project gone bad. Evil science. Somebody stapled another leg on. Yeah. So we can look through these and we can start trying to figure out what's the problem. What could have caused this? We need to create a falsifiable hypothesis. All right, falsifiable means it can be shown to be wrong. It has the potential to be disproven. You can never, ever prove a hypothesis. You can only support it. So here's the deal. Okay. My academic history, by the way, I was never really meant to be a biologist. I always loved science. But my undergraduate degree is in philosophy with religion. And I got biology, because I happened to take a bunch of classes in biology because I thought it was fun. And then I realized I couldn't get a job in any of those other fields. So, biologist. But a common philosophical syllogism, a statement that proves itself. 1, all swans are white. 2, that is a swan. Therefore, the swan is white. But then they found Australia. And Australia messes everything up all the time. In case you didn't know, Australia will kill you if you go there. Australia is pretty much the deadliest place on Earth. There's a box jelly the size of your thumbnail that will kill you before you realize you've been stung. Anyways, super fun thoughts. Scorpions, spiders, dangerous snakes, anything. Yeah, if it's evil. Oh, koalas with syphilis. There we go. And black swans. Not nearly scary now. And black swans! It disproves that whole syllogism. It's no longer all swans are white. So we have to create a hypothesis. And a hypothesis is a statement about how the world works. I want to be very, very clear on this. If-then statements are not hypotheses. If-then statements are predictions that you create from a hypothesis. It's one of my pet peeves, sorry. You're going to create a falsifiable statement. So the genetic mutations cause frogs to have multiple limbs is a hypothesis. The prediction, if frogs have genetic mutations, then they will have multiple limbs, is the prediction that you will test from that hypothesis. Do you see the difference? There is a difference. So when you're writing hypotheses, do not write if-then statements. I know you were originally taught to do that, and they taught you wrong, and I'm sorry. Welcome to real science. You're going to test those predictions using a controlled experiment. An experiment or test will be conducted to gather all this stuff. So how are we going to test this genetic mutation? How are we going to test our hypothesis, genetic mutations cause frogs to have multiple limbs? All right, so we're going to do frog breeding. No, we could totally do that. No, I'm down with that. We can do some frog breeding. The trick we're going to need to deal with a controlled experiment is we need to really look at our variables. Variables are any part of the experiment that might change during, or any part that might change during the experiment. You want to keep as many variables as you can consistent, so that the only variable that you're looking at, the only variable that might change, is the one that you're interested in. That's called the independent variable. The independent variable is the variable that you as the scientist cause to change. The thing that you are interested in. So that's the independent variable. It is independent. It is the one you manipulate, the one that you change. The independent variable is going to have an effect on the dependent variable. This is the factor in the experiment you're interested in measuring. So, for us, our independent variable, in this case, we've got genetic mutation, frog breeding. How could we, let's see, how can I put this? Nobody's ever come up with genetic mutation as a hypothesis before for this particular exercise. How would you test that? Hold on, let me go to the next slide. Controlled groups and controlled variables. Some variables, anything that you keep the same is a controlled variable. If we look at these glasses, what is the same between these two glasses? The amount of water. What else? The size. The shape of the glass. Probably the temperature. There's all sorts of things that are kept the same. What's different? Yeah, there's the gunk, the dirt, whatever it is. The substance inside. So you have the controlled variables are everything except for that one thing that you have changed. Whatever's in there. The pollution, we'll call it that, sure. So then that pollution is the independent variable. It's the only thing that's different. The dependent variable is going to be the results of what happens with that gunk. And you can create a controlled experiment by incorporating a control group, a group that is, for lack of a better word, totally normal, real life, and a group that's experimental, the one that you have manipulated, the one that you have changed the independent variable on. We're going to be doing a lot of controlled experiments in this course. So, now that we've all got that same vocabulary, we have a control group and we need an experimental group. Are you all good with this frog breeding thing? All right, so we can breed some frogs. We take a frog. Here we got our weird three-legged, multi-legged frog. What do we do with it? Well, we captured it, so we're holding it. You breed it with what? With what? A control frog, so like a normal-looking frog? Let's go over here. We'll pick up a normal frog. So over here, we got a pond full of normal frogs. Hey, look, there's a normal frog. Yay! Hey, check it out. Every frog in this pond has a ton of legs. For those who are going into business, you're thinking, now we've got the new product. So what do? We got a bunch of frogs in this pond over here that have lots and lots of extra limbs, and none over there. Well, they've got limbs. I mean, don't get me wrong. They've got normal limbs.
What I would do is breed the abnormal frog with one abnormal frog and also with one normal frog.
Well, it's kind of fun frog time. All right, so we got 2 abnormal frogs. Do we put them at do we do it on the same tank?
I think you put them at 76 degrees for 3.5 hours.
Okay, that'll cook a frog. All right, so we got well, we're talking Fahrenheit, not Celsius. Sorry. All right, so we got a big aquarium here. 2 abnormal-looking frogs. We put them together. We start. That's frogs mating, apparently. They're dropping down a bunch of eggs. Life's good. The eggs start growing in this tank. What's in this? What's in here? Okay, but what I'm saying is, what did we just drop them into? I mean, it's a tank, but what's in this tank? What kind? Fresh water? Pond water? So just like, swish. Okay. Put them in there. What? We did eggs. They grow up. They're all multi-legged. All right, now what do we do? Okay. They have a much smaller container. All right, so the water from there, 2 normal frogs, put them in, and they're all normal. Yep, totally normal. Oh, wait. I'm sorry. Did you tell me to breed one of those frogs over here?
Well, your control group is 2 normal frogs.
2 normal frogs into the water from that pond. They're all normal. Yep.
Now you want to take one of the normal ones and breed it with the abnormal one.
Okay, I need another tank, don't I? I have to put them both in this hand. Another tank. Where's this water?
That water?
Yeah. Where'd it come from?
We're going to mix that water and that water. But we're testing too many variables.
We totally screwed that up. But we put them in! And they're all weird. All of them are abnormal. Okay, let's chill there. Yes. Wait, and when you have a comment, just start yelling. Otherwise, I'm going to totally miss you. Okay, environmental factor. Okay. Ooh, something in the water. How would we test that? So we took our old hypothesis, and we've started something new. And that's the way science works. Running out of time. I didn't expect that to happen in a 3-hour lecture. Okay, what now? What do we do? I got to figure out this frog thing before we go.
What you would do is you would test frogs from that pond in that water.
I only got one. All right. That's right. They're all weird. Oh, you can't safely assume that yet. What's the last thing you have to do? We've checked in this one with the normal frogs, and we get all weird. And they're all normal. That's the way it works, right? Real life experiment, by the way. I'm not making this stuff up. Well, I'm making this part up, but I'm not like this actually happened. Yeah. Okay, so now what? You said environmental effects, so how can we test? Yeah, you figure out the water quality. All right, I'm going to stop us there for about 3 minutes. You've been working on this, so you guys, I need you to use the scientific method. Figure out the answer, okay? I'm going to give you until 12:08 to figure out the answer of why these things have multiple limbs. So 1.5 minutes. All right, so give me what ideas you just came up with. Anything? There's something in the water, but we don't know what. What's in the water? We figured out it's probably the water. You got the environmental effects. Something's in the water. What is it? It's not. But thanks for trying. Not radiation either. Not an enzyme that's messing with their breeding. Come on, guys. I'll give you another minute. All right, so you've had a lot of fun ideas, and they would take a lot of time to test. Going through the scientific method, what's the first step? Right, and you got that, right? Many limbs. Second step. Question. Question is, why do they have all these things? Third step. No. Research. And what did none of you do? Research. All of you have phones sitting right in front of you that you were just checking. Why didn't you use them? Yeah, but then I said ignore that. Listen to what I say. Listen to what I say. So the idea is you could find out the answers. It's going to save you as a scientist so much time. People have done research on things all the time. So this is about control groups still. Sorry, I missed this slide. You got positive groups and positive control groups and negative control groups. The positive control group is the one you know will definitely work. The negative control group is the one you know will definitely not work. For our frogs, which one would definitely not produce frogs with multiple limbs? The clean water. That's the negative control. And we figured out one that definitely does work. That would be the positive control. Does that make sense? The trick is you need to do some research first.
You could probably find something in some of the city websites about
Exactly.
what water systems connect to that pond.
You would have to do research into that. That's exactly it. The research will tell you your positive and negative controls. Gather your data. We've got two kinds of data: quantitative data and qualitative data. Quantity, numbers. We biologists love numbers. Qualitative data, touchy-feely descriptions. I hate those. Quantitative measurements are what we want. Qualitative works to support quantitative, but in the end, as far as hard science is concerned, we want numbers. We analyze the data, we put it together into a graph or something that we can measure, and we draw our conclusions. We accept or we reject our hypothesis. Now for us, we rejected several of our hypotheses. We finally did some research and we figured out the answer. Did anybody figure out the answer? No, of course not. It was caused by a protist. You guys know the frog life cycle? Frogs go from egg to tadpole, tadpole to froglet, froglet to frog. So as a tadpole, they end up getting this protist right about where their hips are forming. And that protist does spins. And when it spins around, it ends up disrupting the cells that are going to become the legs, which leads to an abnormal number of legs. The more you know. So we've applied the scientific method. We determine whatever limitations we have. We report. We publish. We get it out to people. And we let everyone else know what's going on. Otherwise, if we didn't let people know what's going on, they'd be sitting in a class trying to figure out, why do frogs have so many limbs? Here's a historical example of the scientific method. I know I'm going to go right up till the end. Sorry, I'm hungry too. This guy's name is Francesco Redi. Francesco Redi is a horse-faced gentleman who did some research into the spontaneous generation of life. You see, for the longest time, people thought that living matter came from non-living matter. That is, a leaf landing on water would become fish. That's how we got fish. And they thought this because if a leaf lands on water, you see baby fish darting out from underneath it. Yeah. So, and they thought hay turns into mice. Because if you go into the barn and you pick up a bale of hay, mice come out of it. Thus, the hay is turning into mice. Frogs are born from mud. Because if you look at the riverbanks right in the first spring rain, the mud starts to boil, and these muddy frogs pop out of it. Because, mud turns into frogs. He was a long time ago. That you couldn't tell from the picture? You've got flies are born from rotting meat. When you have meat hanging out, flies are created from it. Oh, and my other favorite is bees come from flowers. Because in the morning you go outside and you see bees just flying out of flowers. So the problem is, how do new living beings come into existence? And he really focused on flies here, and he said, well, maggots appeared on meat after a few days. There's no microscope at the time. They weren't really around, so there's no way to see that they were eggs. But he thought that the maggots came from eggs that were laid by flies. So his hypothesis, flies produce maggots, just that easy. And he looked at, he set up an experiment in which you had several different containers. Some containers were sealed, others were open, and there was meat in the bottom of each container. He created a control group and an experimental group. Now, here we have two groups, uncovered and covered. Which is more like real life? Uncovered. That's the control group. What happens in the control group after a few days, do you think? Lots of flies. In the experimental group, the one that he's changed, not so many flies. So we have the control group, the experimental group. We have several variables that are the same. What are the same? What are the controlled with an ED variables? What are the things that are the same between each? Meat, temperature, exposure to air, size of the container, type, yeah. Location, all these things. They are all kept consistent. The only difference between them is the independent variable, which is the covering. He collected data. This is really just an example of a lecture to show you a bunch of maggots, I guess. He created charts and tables. He looked at them, and he drew conclusions. Wow, that's small. Sorry about that. He restated his hypothesis. Flies produce maggots. He accepted it. His conclusions were that flies lay eggs too small to be seen, maggots found on rotting meat are produced from the eggs laid by flies, and maggots are not appearing due to spontaneous generation. That's what he came up with. Yeah, that's good, right? And he didn't see many limitations, so he went ahead and published a study. And that was the end of spontaneous generation. Everyone said, okay, well, obviously, now that he's shown that flies come from meat, or don't come from meat, that's the end of that. Except it wasn't. Turns out, a guy named John Needham said, no, I think you're wrong. Because when I leave a thing of chicken noodle soup out overnight, it grows stuff in it. You guys have left chicken noodle soup overnight, haven't you? Yeah. Don't eat it the next morning. It's disgusting. So he boiled it. He boiled this chicken broth effectively. Nothing was in it. A day later, it was filled with stuff. Spontaneous generation. So what did Redi say to him? He said, did you cover it? And he said, no. So then this guy comes along, Lazzaro Spallanzini, who was Redi's nemesis, and said, yeah, well, I will. So he boiled it and had a cork in it. The next day, it was totally clear. A few days later, stuff was growing. He said, therefore, spontaneous generation happens. This went back and forth for like 50 years. Anyway, you guys know why this happened? You guys have ever seen, you're a little young, I know. Have you seen corks before? What's in a cork? Holes, right. Bacteria. The nail in the coffin came about 30 or 40 years later by a guy named Louis Pasteur. You probably heard of him, pasteurization. No, he didn't make antibiotics. He killed bacteria. He created the swan neck. So what would happen is you boil the stuff with bacteria in it. It kills all the bacteria off. And then the steam goes out through the vent, but the bacteria get trapped in that little neck. So thank you. So then over time, I'm trying, 2 minutes. He let it sit for a day, a week, 2 weeks, a month, 6 months, and nothing grew until he broke off the top. And the day after he broke off the top, it was filled with stuff. This is what put the nail in the casket, and he's the one who's credited with really killing the idea of spontaneous generation.